in order to replicate the DNA, there's going to be a template that's going to be read by a polymerase, and then a product that gets synthesized by the polymerase. On this particular figure, we're comparing replication with transcription. Okay, so replication, transcription over here on the right. Notice the similarities. Synthesis is always, always, always complementary and anti-parallel. It's always going to be complementary and anti-parallel. Whether you're talking about transcription, whether you're talking about replication, it's always complementary, it's always anti-parallel. So the DNA template in both cases is going to be read, and remember what I'm saying here because this is going to be very important. It's going to be read from 3' to 5'. The DNA template is going to be read by the DNA polymerase in the 3 to 5' direction, 3 to 5' direction, and the product is going to be synthesized in the 5 to 3' direction. Same thing for RNA polymerase. RNA polymerase, the template is going to be read from the 3' to the 5' direction, and it's going to synthesize a product, not shown here yet, but it's going to start synthesizing a product from the 5' to 3' direction. Now, there's going to, for DNA polymerase activity, there's going to be needed a primer. I think of DNA polymerase as it's the conservative, quiet, sort of introverted enzyme. It doesn't like to do things on its own. It needs a little help from its outgoing friend, and its outgoing friend is RNA. RNA is the one that's the life of the party. It's going to get everybody dancing. It's going to get on the dance floor, getting their groove on, everything else. So the RNA is initially laid down before the DNA can come in and be duplicated. So remember that point. When DNA is replicated, there's first a little stretch of RNA sequence that gets laid down by something called an RNA primase. But it's the same. It's the template's read from 3 to 5', and the RNA stretch is laid down from 5 to 3'. Now that the primer is there, new deoxynucleotide triphosphates, the little d, deoxynucleotide triphosphates, can come in and add on to the 3' end. So notice this is growing from its 5' to its 3' end. It's growing in an anti-parallel, complementary fashion. T's are paired with A's, G's with C's, A's with U's. Wait a minute, you told me this is DNA. Well, don't forget, this is RNA. So the RNA primer is actually going to have a U. C's with G's. Now that we're into the DNA section, a T is paired with an A with a T, a G with a C with a G, a C with a G. So that's the way the mechanism is going to happen. And it's going to be polymerized one base at a time. The same thing over here for RNA polymerase. For RNA polymerase, there's no primer required because it's already an outgoing enzyme. RNA polymerase uses RNA as its own primer. So it simply comes in, and there's going to be signals that tell it where to do this, which we'll discuss. But where the T in the DNA, it's going to put an A. Where it sees an A, it puts not a T, it's going to put a U. It's going to put a U. Where it sees a G, it's going to put a C, a C, a G, a C, a G, a G, a U. Whoops, that's a mistake. What's going to happen? We'll talk about it. That's the next thing we'll talk about. You see, DNA polymerase is a smart enzyme. It might be conservative, it might be shy, but it's very smart. Why? Because it has proofreading activity. What that means is that as it's chugging along, reading the template from 3 to 5', synthesizing a product from 5' to 3'. Okay, if we go up here, it's reading the template from 3 to 5', synthesizing the product from 5 to 3'. Maybe it puts the wrong base in. So in this case, instead of putting in a C in, a T was put in. But because this has a 3' to 5' exonuclease or proofreading activity, it can go backwards from 3 to 5', and it can degrade that missing base, or that incorrectly paired base, I should say. So if you look at me, if I'm a template, Okay, I'm a DNA template, and I'll start over here. And my template is going from 3 to 5 prime. Okay, from 3 to 5 prime. If I'm the polymerase, I'm going to want to read that template from the 3 to the 5 prime direction. But as I'm reading from 3 to 5, I'm synthesizing new nucleic acid, but I'm synthesizing it in the 5 to 3 prime direction. So I'm reading from 3 to 5, I'm synthesizing from 5 to 3. If I make a mistake and I need to go backwards, if I'm going already in the 5 to 3 prime direction, what is the backward direction from that? Well, it's from 3 to 5 prime. So that's why this is called a 3 to 5 exonuclease activity. It's like this. It's chugging along, reading the template from 3 to 5, synthesizing the product from 5 to 3. Makes a mistake. It's got to slap the gear shift into reverse, right? So now it has to go backwards from 3 towards 5 prime. It's got to chew up that base. That's the exonuclease activity. It's got to put it back into first gear, let out the clutch nice and slow, and then it's going to go back from 5 to 3 prime and extending the nucleic acid as it's being synthesized from 5 to 3 prime. Now that activity occurs in DNA polymerase. So in DNA polymerase, you'll see in the end that the strand that gets synthesized has correctly paired bases. But that doesn't happen for RNA. For RNA polymerase, remember, it was outgoing, it was wild, it was crazy, but it's a little bit. There's no polymerase, there's no proofreading activity associated with RNA polymerase. You're expected to remember that little factoid. So for RNA polymerase activity, you're going to see incorrect bases present in the resultant RNA species. Is it problematic for the cell? Not really, because the half-life for RNA is fairly short. The half-life for DNA is as long as the cell lives. So, for example, mutations that happen to DNA are going to be the ones that tend to be oncogenic and cause cancer, whereas the mutations that happen in RNA aren't going to really present too much of a challenge or a problem for the cell because the RNA is going to eventually be degraded. We say that then DNA synthesis is high fidelity, and that what that means is that the copy that's made in the end is going to look just like the template. RNA synthesis has low fidelity, meaning that the copy that gets made of the RNA is going to have some differences. It's not going to have exactly the same sequence as the DNA template. So to compare DNA and RNA polymerases, let's look at the following table. You see that the nucleic acid is always synthesized from 5 prime to 3 prime. You see that for both DNA polymerase and RNA polymerase. The only difference is that DNA polymerase is going to synthesize DNA, whereas RNA polymerase is going to synthesize RNA. I think you figured that one out already. What's the required template? Well, for DNA polymerase and RNA polymerase, it's usually DNA. Remember, the template is going to be copied or read from 3 prime to 5 prime. It's going to be synthesized, the new copy, from 5 prime to 3 prime. There's a caveat here, and the caveat is that certain RNA viruses or retroviruses that have to first be reverse transcribed to a DNA copy, they may use RNA as a template initially. We're just going to look at human DNA and go from there. And when you get your microbiology review, you'll get some of the caveats that go with this where you have an RNA template. But for the most part, your template is going to be DNA. The required substrates, well, these are for DNA, your deoxynucleotide monophosphates. Even though your substrate is really the triphosphates, DATP, DGTP, DCTP, DTTP, the triphosphates, in the addition process or the polymerization process, only one phosphate is going to get added. And actually 2 phosphates get lost as pyrophosphate. And that actually helps drive the whole polymerization. That goes beyond the scope of what you need for your board exam. For RNA polymerase, it's just the NMP, or nucleoside monophosphates that get added. So it's ATP, GTP, CTP, or UTP. But once again, it's only adenosine monophosphate or guanosine monophosphate that get added. It's not the 3 phosphates, it's just the 1. And that's what forms the phosphodiester bond. Is there a primer required for DNA polymerase? Well, yes, there's an RNA primer. It could be DNA, but 99.99% of the time, it's RNA. And that's what you should know for your exam. For RNA polymerase, there's no primer required. No primer required because RNA is its own primer. Is there proofreading activity, this 3 to 5' exonuclease activity? The answer is yes for DNA polymerase, whereas the answer is no for RNA polymerase. No primer required. Therefore, transcription is much more error prone than is replication.